Hello, ACA biology students. This is Ms. Sheely with your Chapter 5 video lecture notes. I first have to apologize for it taking so long. I went to record it today during B3 and we had a fire drill, so it didn't work. All right, so today's video lecture is on the structure and function of cell membranes. So first we should just review the general function of cell membranes. There's 6 major functions of the cell membrane or plasma membrane. They hold the cell together. They control what goes in and out using different processes such as diffusion, osmosis, and active transport, as well as others. We'll go into those in a little bit later in the lecture. They protect the cell in general. They allow the cell to recognize and be recognized. So this is cell-to-cell signaling or even cell to foreign invaders like an immunity. They also allow for binding to other cells, so cells actually joining to other cells or other molecules, and this is really beneficial in tissues and such as your skin or muscle tissue. They're also a site for some biochemical reactions, so there are some enzyme reactions or areas for reactions that can occur in the cell membrane. So let's first review from last class and homework before that about lipids and phospholipids. So the cell membrane is a phospholipid bilayer. Phospho mean phosphate component, lipid being fat, and bilayer mean 2 layers. So remember that a single phospholipid is amphipathic, which means it has a hydrophilic and a hydrophobic part, with the phosphate head being the hydrophilic part and the hydrophobic and the tails being the hydrophobic part, and that those tails can be saturated or unsaturated. So saturated would have only single bonds and unsaturated would have at least one double bond, whether that be a cis bond or a trans bond. So when you if you were to drop some phospholipids on top of water, they would form like in this image down here with the hydrophilic phosphate heads touching the water and then the hydrophobic tails just kind of being upside down or floating away from the water. If you were to submerge them, they would form a micelle, which is where you would have water on the inside. So this is if you were to lay them on top and then submerge them, so they would form with their tails all pointed in. So this on the inside of a micelle is no water, okay? So this is all open space or air, and the tails are happy away from the water. If you were to submerge them, not head first, but just drop a whole bunch and submerge them into water kind of willy-nilly, they would form spontaneously into what we call a liposome. And that liposome, it would be the phospholipid bilayer, and you would be able to see the water on the inside and the water on the outside. All right, so let's go through the history of the cell membrane model first. So we were able to identify the plasma membrane as a structure in the 1890s with the use of microscopy. About 25 years later, in 1915 timeframe, we were able to identify through the process of several experiments that there were 2 major chemical components to the cell membrane or plasma membrane, and that those 2 components were lipids and proteins. But we didn't have any idea how they were arranged, and so there were lots of scientists doing lots of tests. But the first proposal of the structure was in the 1930s, particularly around 1935, and a team called Davison and Donnelly proposed the first major model, and it was a modification to a previous model. And this was the first time where we said that, or they said that the cell membrane is a lipid bilayer, and that it was coated on either side with a layer of globular proteins. So those are just proteins in any random shape. They also said that this protein coat had no particular structure and was simply formed by the adsorption from the solution. They had no proof or anything to justify this. This was just their thoughts. There were lots of people who had lots of thoughts, But in the 1950s, we established electron microscopy. And with electron microscopy, we were able to see details of cellular structures that we had not seen before. And when we looked at cell membranes and cells under an electron microscope, we were able to see, just like this image here, this is a electron micrograph image of a cell, that the cell membrane looked like it had a railroad track appearance. And we knew at this time of electron microscopy that proteins showed up very much darker than lipids did. So this railroad track appearance really did support their idea and their theory, their proposal of this structure of the lipid bilayer being sandwiched or coated by globular proteins. And this idea went on for quite some time. There were lots of, or several different experiments that started to disprove this idea. One of the ones that I like the most, it's not something you're responsible for knowing, is this idea of freeze fracturing. So they would freeze cells and then they would fracture them along that inner, so along, try and zoom in here, along the inside of the membrane. And when they separated the two layers of the membrane, they found that the proteins were not on one side and the other. They actually went all the way through. So this piece of evidence, as well as some others, led another team, Singer and Nicholson, to publish an article in Science in February of 1972. And their article was called "The Fluid Mosaic Model of the Structure of Cell Membranes." And they were the first to propose this idea of the cell membrane being a fluid mosaic model. They said and thought that proteins occupy a variety of positions. This was supported by that freeze fracturing technique that I mentioned just a moment ago. They were also the first to name the proteins based on their location. So they termed the small proteins peripheral proteins, and they said that these peripheral proteins were attached to the inner and outer surfaces. They also termed the integral proteins and defined them as being embedded in the phospholipid bilayer by protruding out either one side or spanning the membrane of both and protruding out both sides. They also said that both the phospholipids and both types of proteins are free to move about the membrane itself so that it was fluid. And so they put these terms together and then they labeled the cell membrane the fluid mosaic model, or they labeled their model as being the fluid mosaic model of membranes. They also said that cell membranes were about 5 to 10 nanometers in thickness. All right, so let's go back to what a phospholipid bilayer is, or what the fluid mosaic model is, or go into that in a little bit more detail. The fluid mosaic model tells us that the structure of the membrane is flexible and adaptable in motion, but it's not a solid, and it's not fragile like the skin of a balloon. So if you were to stick a needle into the skin of a balloon, you would puncture the balloon and it would pop. But if you were to do the same thing and stick a molecule or a needle into a cell membrane, it would not pop. So I actually want to take a second and show you this video. This is a technique that's used in vitro fertilization, but it's a really good example and visual of how the cell membrane does not get punctured.
This video is prepared by my colleagues Dieter Egli and Kevin Hagen. And you see here on the left is what's called a holding pipette, which gently sucks on the egg. The egg is surrounded by a membrane called the zona. And you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out, and then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. So if we could start the video, please. You'll see now that this drilling pipette is going to drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part. This pipette, you'll note, isn't really sharp like a syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus. And if you look carefully in the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up. So that nucleus is going to be squirted out now because we don't need it anymore. And there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you two, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side, that it's held by this holding pipette on the left. There's drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. The pipette goes in, and these pipettes are operated with a piezoelectric device. So you can't see it here, but it's like a little jackhammer going very quickly, like a Woody the Woodpecker, getting in there to then squirt the nucleus in. Here we'll see it again, a little hole, and the zona is prepared, and now the nucleus is going to be squirted inside. So there's two examples of what's called somatic cell nuclear transport.
All right, so you could see that the individual phospholipids had some freedom of movement, that the cell itself did not pop like a balloon when it was punctured by the pipette. So it's actually very difficult to burst or tear a plasma membrane because the fluid properties of the phospholipid bilayer allow the membrane to reform very, very easily. All right, so let's go through the names and functions of the parts of the cell membrane. So I'm going to start here with the integral proteins. So integral proteins could be, they could go all the way through, as this purple one does, or they could just go through one side or the other. They could be enzymes or pumps. You also have integral proteins such as this channel protein over here. This one does span as well, and these guys would carry the molecules through the plasma membrane. You have smaller proteins such as this peripheral protein, and so they're going to be just on one side. It could be the extracellular side, such as this blue one, or on the intracellular side, such as this pink one. And they're going to act as receptors, or they could recognize other cells. You have two different carbohydrate structures. You have one that's shown here called a glycoprotein. And these glyco, the prefix glyco refers to the carbohydrate chain. So these little ball and stick things are the carbohydrate chains, and they are attached to proteins. So we call this a glycoprotein. And this particular one is a chemical receptor. These carbohydrate chains, though, could also be attached to a phospholipid head, and then they would be glycolipids. Collectively, the glycoproteins and glycolipids are referred to as, or collectively they're called, the glycocalyx, and are referred to as being the sugar coating of the cell. This is the area that viruses bind to and thus how they attack a cell. So the last structure that we're going to talk about, I'm skipping over the phospholipids and their components because we've already talked about those, is the cholesterol. And remember from our discussion and our notes on lipids from your 3.3 section, cholesterol is a type of lipid, and it affects membrane fluidity at different temperatures. So the fluidity of the membrane, the fluidity as your temperatures in your cell or the cellular environment decrease, the saturated fatty acids will start to become dense or more tightly bound to each other in the membrane, and this will solidify the membrane. But when an unsaturated fatty acid, so remember an unsaturated fatty acid would have that double bond, usually in that cis formation. When an unsaturated fatty acid compresses due to a decrease in temperature, those kinks will actually maintain a space between the neighboring phospholipids, so the membrane is able to stay fluid. So the membrane itself, just with the phospholipids, some of them being unsaturated, will be able to maintain some of their fluidity. But cholesterol adds to this. Cholesterol prevents both lower temperatures and higher temperatures from changing or inhibiting the fluidity of the membrane. So it's going to decrease that solidification because it's going to space out those phospholipids. But also if your temperature increases too much, they might move too much, the phospholipids, so it's going to actually, the cholesterol will help keep those phospholipids from being too fluid as well. All right, so these are 6 functions of membrane proteins, both peripheral and integral. It could be a combination of these. This is not covered in your textbook, but it is something that I find very important, so I wanted to bring this to your attention. So the first thing is just a regular channel protein. So this channel protein would be open all the way through in both sides. This is a type of passive transport that we will get to in a little bit. Another would be an enzyme. The enzyme could be on a large integral protein or it could be on a small peripheral protein, but this would be an area where there would be a reaction occurring, and this would be either on the interior or exterior cell membrane side. Another thing is that proteins could be hormone receptors or surface receptors where they are open for a signal, whether it be from a protein or another molecule, and then that will stimulate an action on the inside of the cell. Some proteins could be pumps, and we will talk about pumps for active transport a little bit later. Some proteins can be used to adhere cells together, so they're adhesion proteins, so binding cells together. And then again, I just talked about the glycoproteins being part of the cell-to-cell communication, but you can also have glycolipids do that as well. All right. So that was it for the first section. So the next section we have is 5.2, and this is passive transport within the cell membrane. So the phospholipid bilayer is what we call selectively permeable. So this is a controlled entry and exit of molecules. Some molecules can go through easily, which would be diffusion. Some go through with a tunnel or with help. That would be facilitated diffusion. Some need energy to get them through. That would be active transport. And there are larger molecules that can't get through with any of those, and they need their own membrane to get through. And this would be an example of endo and exocytosis. So remember that the polar heads are attracted to other polar or charged molecules, but the nonpolar tails will repel any charged molecules. So many ions cannot pass through the membrane without help. All right, so let's review solutions. So remember that solutions consist of solutes dissolved in a solvent, and the concentration is a measure of the amount of solute in a solution, and thus the measure of the solvent as well. So if you have a pure water, which is no solute, then you have 100% water. It's not a solution, such as the cell on your left here. This dotted line in the middle is representing our selectively permeable membrane. On the cell on the right, we have a 50% salt solution on the left side of this cell and a 20% salt solution on the right side of the cell. So we have, if you have 50% salt, then you have 50% water. If you have 20% salt, then you have 80% water. So the solutes and the solvents will move back and forth. So diffusion is the passive net movement of molecules from regions of high concentration to low concentration. Passive means no energy. Net is the overall movement. And remember that this is overall movement because molecules are moving all the time. And the higher the temperature, the faster they go. The lower temperature, the slower they go. And then from high to low concentration means down a concentration gradient. I like to think of this as a sliding board or a slide. And when you are at the top of the slide, you will sit down and you can put your hands up and you don't have to do any work to get to the other end. So that's going down a gradient, so going from the high end to the low end. Diffusion can occur through a partially or selectively permeable membrane or from regions of just high concentration to low concentration. So if you think about that example, not a cell, but if you just had a beaker of water and we put food dye in this, and then what would happen? The food dye would, over time, mix without any stirring needed. All right, so I want to watch this little video of how diffusion works.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, And even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
All right, so if we look at this slide, there are a few different factors that affect diffusion, and here is one. So the effect of a diffusion, the speed of diffusion is going to be affected by the concentration gradient. So for example, we have a 70% salt solution and a 20% salt solution. So the difference between 70 and 20. And then over here on the right, we have 50% and 20%. So that difference being between 50 and 20. So this difference of between 70 and 20 is much greater than the difference between 50 and 20. They'll both move from the higher side to the lower side, but this one on the left will move at a faster rate because the molecules will have more energy and will move more quickly. There are 6 other factors that affect the rate of diffusion. One is the mass of the molecules that are diffusing. So the heavier molecules diffuse more slowly. So therefore, they move more slowly, therefore they will diffuse more slowly. Temperature, I already mentioned that a little bit earlier. The higher the temperature, the faster the molecules will move, and thus the faster the rate of diffusion. The solvent density. So solvent for us in biology is typically referring to water, so we're not going to change our density. But if we did look at other solutions, we would have a change in our diffusion rate. So as the density of the solvent increases, so as we get higher density or more weight, the diffusion rate will decrease. And we can get this with dehydration. So dehydration in your cells can actually lead to a decrease in fusion rate within your cells. Another one is solubility. So nonpolar and lipid-soluble materials can get through the membrane a little bit faster. So they're going to allow for a faster diffusion rate because they can get through that tail area. The amount of surface area and the thickness of the plasma membrane. I mentioned earlier that the typical thickness of a plasma cell membrane is between 5 and 10 nanometers. The thicker membranes will decrease the diffusion rate, and the more surface area you have will increase the diffusion rate. So your nerve cells with all the dendrites will have a faster diffusion rate, as well your microvilli and your small intestines will have a faster diffusion rate. And the last thing we have is just distance traveled in general. So the greater the distance that the molecule must travel, that the slower the diffusion rate will be. So this is really just talking more about putting a limitation, or there being a limitation on cell size. All right, another type of passive transport, is facilitated diffusion. So facilitated diffusion is still the passive network of movement of particles, still going from regions of high concentration to low concentrations, still through a selectively permeable membrane, but this time we're facilitated or helped by carrier proteins, and they could actually be carrier proteins or channel proteins, just proteins in general. So some molecules cannot get through selectively permeable membranes. It depends upon the properties of the molecules. If they cannot get through, then in order to get through, they would need help. So you have carrier proteins and channel proteins, which would be integral globular proteins. So these are ones that could span the whole width of the membrane, going from protruding to both sides, or just being on one side and then able to move through and carry the substance. These channel proteins are very specific to their molecules, and this is determined by the polar regions of the proteins that would be on the inside and would react to these ions. We'll get to more of that later. This is down the concentration gradient, so down the slide, so therefore still not requiring energy. All right, some of our channels are just always open, and some are what we call gated, where they open on one side and then close on the other. So I do want to watch another video about how facilitated diffusion works.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
All right, another type of passive transport is osmosis. So in osmosis, we're still looking at the passive net movement of molecules, but this time we're looking specifically at water molecules. This is a region of low solute concentration to high solute concentration through a partially or selectively permeable membrane. Solute materials are not going to pass, these solute materials are not going to pass through the membrane due to their properties. So we're looking at the movement of water. So osmosis is still passive because it's still down a concentration gradient, because we're not talking about the movements of solutes. We're talking about the movement of the water molecules. So in this case, if we have 50% solute, we would have 50% water. If we had 20% solute, we would have 80% water. So this would be the higher water, and this is the lower water. So the water would move from the right side to the left side. All right, so here's another video of osmosis.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
All right, so those were some terms right there at the end about being having to do with what we call tonicity. So tonicity, if you take a normal cell and you place it in a hypertonic solution, the water will leave the cell because there is more, the water concentration is greater in the cell than it is in the hypertonic solution. So if you have an animal cell, such as these red blood cells at the top, that cell would shrivel and shrink. If you have a plant cell, that plant cell would also shrivel and shrink, but the cell itself would maintain the same size because of the cell wall. Now you can see here that the cell membrane is still attached at a few random places. So now we have these giant pockets of air in the cell, and we call this is called being plasmolyzed, or having gone through plasmolysis. Now, if a cell was placed in an isotonic solution where the water concentration is the same in the exterior environment as it is on the interior, water would still leave the cell, but it would also come into the cell. So it would go in and out in a nice, even, equal balance. And you would have nice, normal, healthy cells in an animal cell. In a plant cell, we would still not be full, super full. So we actually call this being flaccid, just normal and regular. Now, if a cell was placed in a hypotonic solution where the water concentration was more pure or higher outside the cell than inside the cell, then the water would go into the cell. If this was an animal cell, the amount of water entering the cell will cause the cell to swell and could cause it to burst. In a plant cell, that plant has a cell wall. So that cell wall is not going to allow the plant cell to burst, and instead it's going to get just very, very full, and most of the water will go into the vacuole, and this is going to be what's called the plant cell being turgid, or very, very full of water. All right, the last thing I want to talk about in this passive transport and movement of osmosis is a particular protein channel called an aquaporin. And this is a type of facilitated diffusion. So some water can move in and out of cells on their own, but water, being a polar molecule, moves very slowly through the cell membrane because of the hydrophobic tails. So in 2003, two gentlemen were co-awarded the Nobel Prize in Chemistry for their both of their work with the aquaporin and the ion channels. So Dr. Peter Agre was awarded his half for the discovery of the water channels, and then the other half went to Roderick McKinnon for the structural and mechanistic studies of just ion channels in general. So I do want to play you just a quick 3-minute video interview with Dr. Agre. He did his work on frog embryos. So I'm going to play you this video at this time.
My name is Peter Agre. I'm director of the Johns Hopkins Malaria Research Institute at the Johns Hopkins Bloomberg School of Public Health, Baltimore, Maryland, USA. Some ways I think I'd say research chose me. I was a student in medicine at Johns Hopkins back in the 1970s, hoping to do some research relevant to the diseases of the third world. I was involved because of the humanitarian interest. I guess there was a threshold I must have passed where I became a scientist. It was not a one-way street at which I would end. It was science from here on. Being from a large family, I'm used to talking to people, sharing information, getting ideas. And I talked to scientists. I talked to dozens of scientists. Explained that we had a new protein. We thought it was a channel protein from the red cell membranes. And we always got the same response. Red cells don't have channels. You must be wrong. Until I finally talked to a colleague, John Parker, at the University of North Carolina at Chapel Hill, a very dear friend, a hematologist, a blood specialist, who was also a physiologist. And he immediately recognized red cells, renal tubules, roots of plants. These tissues have one shared property. They're highly permeated by water. Maybe this is the long-sought water channel. It was before our first public lecture. My postdoctoral fellow, Greg Preston, wonderful fellow, charming young man. We were all having lunch, but he had to give the lecture, so he was the only one not to have beer. So we were thinking we need a name, a functional name for these water channel proteins. And there were 6 of us or 7 of us at lunch. And no one remembers exactly where the name came, but we were thinking of names like maybe hydropontin or hydrophorin. Someone else thought maybe aquaphorin, or the Greek aquaporin. That's it. Everybody said, that's it, aquaporin. The generation of pure water is theoretically possible with the aquaporins because this is a water selective filter. Only water can go through this filter. The engineering of this, though, has some technical difficulties because the native membranes are very tiny, and so only nanoscale water purification can occur. A lot of individuals got very excited, attention from around the world, even a Nobel Prize. But, you know, I honestly don't see myself as Einstein reincarnate. I see myself more as Huckleberry Finn. Happy-go-lucky individual with a good team of scientists and friends looking for adventure. And boy, did we find one.
All right, so what Dr. Agre and his team actually did is really cool. They looked at, they injected the complementary RNA strand into these frog eggs. And these frog eggs are very small and known to be quite impermeable to water. If you think about frog eggs being laid in water, they need to be impermeable. They don't want to take on the surrounding environment. And you can see here these top eggs, they were not expressing the aquaporin protein, and that protein is abbreviated AQP1. And then here at the bottom, you can see these proteins swelling and joining, and that's because they are expressing that protein. So they did find that the proteins were not found in all cells, but the ones that they are found in, they do allow for water movement to be much, much higher. And we'll actually look at an aquaporin molecule in class. All right, so that's it for 5.2. So we're halfway done with the chapter. So the next section is 5.3, which is active transport. So active transport uses energy in the form of ATP to move the molecules against a concentration gradient using a protein pump or a carrier protein system. So these molecules, remember, not all molecules can pass through the membrane due to their properties, and sometimes we don't want to go down the concentration gradient. We want to go up, so we want to climb up the slide. So active transport is a key process in homeostasis in organisms because it can really help to reset a cell after an impulse has been set. So we're going to look at this video. Okay. This one, keep it. All right, so this video does not have any sound, but we're going to look at 2 parts to it. So the first part is actually primary active transport, and the second part over here, this protein, is involved in secondary active transport. So again, in primary active transport, we're creating a differential in charge that's dependent upon ATP. This particular molecule is the sodium-potassium pump, or sodium-potassium ATPase. So if you notice, there are some sodium ions joining and some potassium ions joining. And after 3 sodiums, you can't see them because they're stacked on top of each other, and 2 potassiums join, then the ATP comes over and binds. And when the ATP binds to the sodium-potassium pump, that allows the molecules to switch in reverse direction and be released. And then you'll notice here that our ATP, adenosine triphosphate, has split into ADP and a free phosphate ion. And then our sodiums are released to the extracellular surface, and our potassiums are released to the interior. And this will be repeated. 3 sodiums, 2 potassiums, ATP coming in, switching ADP and a free phosphate ion going back. This will happen one more time. And now what you'll notice is that we have an influx of sodiums on the outside or an influx of positive charge due to the increased number of sodiums on the outside and a decreased number of positives on the inside because we've swapped out at a 3-to-2 ratio. So now we have a charge imbalance for where the cell wants to be. So now we'll use the secondary active transport, and this particular one is called a symporter, and we'll go through all three types of carrier proteins in a minute. And a secondary active transport system is movement in response to a charge differential that's created always by a primary active transport system. So in this case, we're going to bring sodium, and once sodium binds, then a glucose can bind, and we'll bring them both to the cell. And then this will repeat until our charge differential is reset. All right, you do need to know the details of sodium-potassium pump and how that works. Protein pumps in general, not just sodium-potassium pump, but all protein pumps are specific to the molecules that they're going to move. The molecules will bind to an active site inside the pump, and then the release of the energy from the ATP will cause a conformational change in the shape of the protein pump. So the last animation was really good at showing the overall structure, but it did lack a lot of key essential details on how the protein pump worked. It oversimplified that. So I do want to show you this video that will go through that in more detailed and correct information.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
All right, and then just to give you a breakdown of what happens without any movement, there are 4 parts to the movement of the sodium-potassium pump. So the sodium-potassium pump is always open to the inside of the cell. Then the 3 sodiums will bind to the inside of the cell. Once the 3 sodiums have bound to the inside of the cell, that activates or opens the active site on the side for the ATP to bind. When ATP binds, then the protein pump will have a conformational change and open to the outside or extracellular space. Then the sodium will be released. It will stay open to the outside until the 2 potassium ions bind to it. Once the 2 potassium ions have bound to the inside, that will release the ADP and the free phosphate ion from the pump on the interior space, and that will cause a conformational change where it will open back up to the inside. And that process is just repeated and repeated until that balance is no longer needed. All right, so with our carrier proteins, we have 3 different types of carrier proteins. We have carrier proteins that move 1 molecule in 1 specific direction, and that's called a uniporter. We have a symporter, which is going to move 2 different molecules, but in the same direction. And then you have an antiporter, which will move 2 different molecules, but in two different directions. So the one that we saw that was the secondary active transport with the sodium potassium pump a couple videos ago was a symporter. All right, the very last section of the textbook is 5.4. This is bulk transport. The two different types of bulk transport that the cell uses is exocytosis and endocytosis. Exocytosis is the export of macromolecules from the cell, so large molecules. This is not just active transport or anything like that. Endocytosis is the import or bringing in macromolecules. So exo, exit, endo, enter. Endocytosis actually has two subtypes. You have phagocytosis and pinocytosis. So phagocytosis is referred to as cell eating. This is large particles, for example, a white blood cell engulfing a bacteria that has invaded your body. I do want to look through this very, very, very simple animation. Doesn't quite show everything we need. So we have our little bacteria, our cell membrane comes up around, and then we have a vesicle that will then join with a lysosome. Let's watch that one more time. So we have our bacteria, our large molecule. It's being engulfed by the membrane, creating a vacuole. That vacuole will join with a lysosome for breakdown of that molecule. What that video didn't show or didn't include quite so accurately, as it is a very simple video, is when as that vesicle is forming, it's actually also going to have a protein shell around it for stabilization. And those proteins are called clathrin proteins. And those clathrin proteins will stay around the vesicle until it is fully formed and enters the cell. And then the clathrin proteins will disassemble from the vesicle, and the vesicle will form with the lysosome. Pinocytosis is referred to as cell drinking, and this is the ingestion of molecules and fluids, smaller than the phagocytosis, but still too large to enter any other way. This was actually discovered and named in 1929 by Dr. Warren Lewis, and he thought it was just fluid only, which is why he termed it a little bit differently. Although it does have a slightly different process, the overall process is very similar. So here you have a solutes outside. We're going to have the ingestion of these molecules through a vesicle, but now we do not need the lysosome to bind with it. So we have the cell membrane forming around, forming a smaller vesicle, and then there's no need for a lysosome. Sometimes the cell needs to move molecules from one side of the cell to the other, and that is a process called potocytosis, P-O-T-O-cytosis, and that's going to use a protein similar to the clathrin proteins. They're proteins called cavelins. And again, they will form a vesicle. It will be surrounded by these proteins called cavelin proteins, and then will transport the molecules to the other side of the cell where it will release them. And then the exporting of materials is exocytosis. So here's a really good example or step-by-step picture of how exocytosis is formed. So exocytosis is expelling materials from the cell. So this could be any type of waste material that's in the cell. It's first going to be enveloped by a membrane to form a vesicle. That vesicle will then fuse with the plasma membrane interior. And here's exactly how that fusion happens. So the vesicle will come up. So here's the vesicle, and here is the plasma membrane. They're going to get really close together. And then remember, these guys are fluid, so they're always moving. And they're going to join together and begin to fuse. So they're flowing around each other, but you can see we have one very thick layer, and then that becomes much thinner as they continue to move. So we have a single small bilayer called a temporary intermediate structure. You're not responsible for that information. But then that will continue to separate and bend as they continue to move, and this will create a pore that opens. And so our cell membrane is never, is never exposed to the outside environment, so we can release our waste that way. There's never an unbroken section of the bilayer, and you can see this a little bit better here and not on the blow up. It was really, this is really amazing. Actually, this process wasn't discovered, the method for this wasn't discovered until 2002 by 2 researchers using X-ray diffraction images. So we are still discovering stuff on a regular basis. All right, guys, whew, that was a lot of information. I hope some of it was review. We will continue to dive into this in class. I've got some models to show you, and we'll do some activities to work through it as well. Okay, look forward to seeing you in class.